Thank you so much, Eric. Very well said. Good morning, everyone, and I'm Grace Anderson with my colleague Mauricio Lorenes, our presenter for today. And before we start, I would like to give you a warm welcome and to say thanks for being with us and for giving us the opportunity and invitation to discuss MST, or also known as DNA source identification. Our presentation today will provide you an overview about the current methodology that are being used by local governments, watershed managers, and environmental engineers to help them control the fecal contamination sources and still meet the water quality standards. So during the presentation, like Eric said, you may have questions or issues related to your individual water quality programs, and we will try our best to address all of this. So I guess we're all ready, we're all set, so please go ahead and start, Mauricio. Thank you.
Okay, thank you. I'm very excited to present today. So background on our company. We were founded in 2002 to bridge a gap between a lot of really interesting research that was going on at the time and a lack of implementation. And over the last 12 years, we've become what we consider the leader in commercial microbial source tracking services. So last year alone, as an example, we did over 70 source tracking studies, and we're very active in research and development, collaborating with the state of California on the development of their source identification project, also participating with on the US EPA method standardization study, and collaborating with what we'll talk about later on digital PCR validation with SCORP. We've licensed technology from the US EPA on human, cattle, chicken, and dog, and also have 9 additional sources that we can detect. And we have a very strict quality control and quality assurance procedure, and through that we're in the process of finalizing our ISO 17025 accreditation, which time will be the only accredited laboratory in the country to do microbial source tracking. That's a little bit of background of our laboratory and our role in source tracking. And so the presentation is broken into 4 parts. The first part will be a little bit of background on the method and those new developments we had talked about in the presentation subject, applying microbial source tracking. So this is going to go over really practical overview of what things you need to be considering when using this technology. Then moving on to case studies, 3 main case studies that we were involved in over the last few years. And then a conclusion period where we'll be able to have discussions and you can ask questions on any of the slides. So the problem that microbial source tracking is trying to address is the introduction of fecal pollution into a watershed. And so monitoring is occurring to see if there is, in fact, fecal pollution within the watershed. The challenge is that the introduction of the water fecal pollution is not obvious. Sometimes it could be coming from household waste, other times from agriculture, other times from industrial waste. And it's not always obvious what is the cause of that fecal pollution. And so regular, traditional fecal indicator methods, you take the water sample, you enumerate it, and then it tells you you have general fecal indicators in your water, which is helpful for monitoring and regulatory purposes. However, it doesn't really provide you insight into what's causing that pollution. And so over the last nearly 30 years, this has been an emerging and very active area of research across the country and internationally. How do you get better insight? How do you do it better than just telling somebody you have pollution? And a lot of resources have been put into this question. And as of today, the current state of science is the recovery, filtration, isolation of DNA in a sample. Then that DNA is put into a machine. And then we look for host-specific indicators from each animal that you're concerned with using qPCR to tell more specifically not only that you have pollution, but what are the host sources causing that pollution. And so that's the intent of microbial source tracking methods. This gives you sort of the rundown of the last 30 years. Where are we today? The vast majority of laboratories are using this qPCR-based approach to tell what are not only general bacteria levels, but also what are the hosts causing that. I'm not going into the science too much on this presentation because there's been other presentations that really go more deeply into how all of this works. But what you need to know as a practitioner is that you will get an idea of what is causing the pollution. So this sums up 30 years. Now, what has happened more recently in source tracking that's new and interesting and causing the latest advancements in source tracking? So there's three main developments. One is digital PCR's introduction into environmental water monitoring. So this is a technology borrowed from the clinical environment, and I'm actually going to show a short video that was created by Biorad, one of the digital PCR vendors, and Scorp. And so I'm going to pull this up now, and hopefully each of you will be able to hear and see this presentation.
The importance of monitoring the ocean for human impacts is multifold. In the case of my department, we're interested in using the most advanced methods to protect public health from pathogens that may come off the land.
Our hope with this new droplet digital PCR assay is that this becomes a valuable tool for monitoring agencies so they can reduce the variability in their results and lower the chances of having false negatives.
Scorp is the Southern California Coastal Water Research Project. We're a local government agency that does independent research on human impacts to the ocean. So the primary way that bacterial contamination reaches the ocean is through the storm drain system. And here in Southern California, you notice everything is paved over. Everything's engineered to prevent flooding. So when it rains or when there's runoff, when a fire hydrant breaks, it all runs down to the beach just as fast as it can.
So the main concern is public health risk due to human fecal contamination getting into water. So in order to protect public health, the waters are usually monitored regularly for fecal indicator bacteria such as Intercoccus. However, the current monitoring method for Intercoccus is culture-based, which is slow, takes a whole day to get the answer. So what we're trying to develop are genetic method that's fast and specific to organisms we're interested in. So what we usually do is we take water samples from the beach, storm drain or creek. We bring it back to the lab, we filter them to capture all the microorganisms on the filter. Then we extract DNA from the filters. Now the genetic material from the filter will be available for quantification by qPCR or digital PCR. Last year, EPA just proved a qPCR-based method for monitoring Intercoccus. But qPCR has its own shortcomings. Number one, it relies on external standard. Depending on where you get your standard, your answer could vary by half a log. The second thing is that because the environmental samples we get are very complex, they often contain chemicals that interfere with the qPCR chemistry, leading to false negative, which is very undesirable for public health protection. So there's three reasons why we're interested in droplet digital PCR. One, of course, is provides absolute quantification without the need for external standard. The second thing is that digital PCR is more robust against inhibition. The third thing is it allows multiplexing, which means you can measure multiple organisms at once. So the assay we have developed using droplet digital PCR can measure Intercoccus and human fecal bacteria daily at the same time, getting information on general fecal pollution and human fecal pollution at the same time at a lower cost. So the results we're getting with the droplet digital PCR are more reproducible than qPCR and also more robust against inhibition. And so we're very happy about these two. That's what we set out to do.
So our goal is to bring the best methodology for testing water to the entire country. Well, what we'd like to see is all the monitoring agencies replacing qPCR with the digital droplet PCR. And the reason for that is we feel it's going to be more accurate and better protect public health.
So with the droplet digital PCR, it's really exciting. It's a technology for the future, and it really opens the doors to a lot of research questions we were not able to answer before. So a lot of new projects are getting started because of this technology. So it's fun, it's very exciting.
So as you can see, SCORP has really put a lot of effort behind this digital PCR initiative, and we were excited and fortunate to have worked with SCORP on an interlaboratory validation of the technology. And so Yiping, which was shown in that video, we had been working with her, and she was interested in seeing how well this technology works across different laboratories. So we took the initiative to receive a loaner for 6 months, and we did a comparison study saying, can we reproduce the same results that SCORP can produce in their laboratory? Is there consistency with data, which can be a challenge with qPCR? And there was. And so we will be, hopefully in the next year, publishing the results of that study. And because of the upfront effort we've made in trying digital PCR, we have now the ability to use and offer this technology on a trial basis to a few key projects this year. And so this would be really interesting for larger projects because of the upfront investment and in using this technology. And it's also interesting for people that deal with wet weather events that are prone to inhibition. Digital PCR can potentially overcome that. And so digital PCR is the first development, and we're very excited to be involved with the introduction of digital PCR in the environmental industry. So the second development recently was the state of California's release of an official Microbial Source Tracking Identification Manual. So this was created, again, by SCORP for the California Water Resources Board as a way of starting to set up a framework of how you actually apply microbial source tracking in real-world water implementation projects. And so this study is a tiered approach to doing source identification, and we'll go into some of the themes from that study later on. But what was really interesting is that this was the first time really that there was scientific consensus across a series of 27 different laboratories throughout the country. And everyone said, you know what, there are methods, these methods work. And everyone was surprisingly and amazingly in agreement. Our lab was a participant in this study. And so based on this research, there are now a series of series of methods for source identification that can be used in the state of California and sets a precedent that source tracking is really a mainstream tool now in California rather than strictly a research tool. So that's a big paradigm shift from just a few years ago, where this is really just an experimental technology. And this is a technology that can help people now on real-world problems. This third set of developments has been the involvement of the US EPA in microbial source tracking. And the two key developments have been the US EPA's invention and patenting of methods to detect human, cattle, and dog. There's another test for chicken, but it's not as commonly used. So what does this mean? This means the US EPA, their research branch, went out and invented a way of developing source tracking tests. Then they patented that, and then they invented source tracking tests for these three sources. They patented it, and through relationship we have with them, we acquired license to use these three tests on implementation projects, real-world watershed management projects. So that's development one with the US EPA. The second, and actually a separate development, is that the US EPA is finally going to create an approved source tracking method. It will be based on qPCR, and they've already gone through the three most significant steps, which is method selection, figuring out is it qPCR, is it next gen, is it library methods, doing their technical evaluation, then doing a method validation, creating these operating procedures and quality control measures, and then doing a multiple lab study to make sure that these tests actually work. Again, our lab took part in this validation. So that's where we are today. The final step is for a decision to be made by the EPA Office of Water to approve this method, and then that will enable the EPA to start using source tracking for EPA policies and programs. So this is going to make a huge impact on how source tracking is used in the very near future. So those are really the biggest developments just in the last 1 year. And this field is moving very rapidly, and you need to be constantly staying on top of what is the latest set of methods, what are the latest developments. And so now we're going to go to the third part, which is how to actually apply microbial source tracking on a project. So the first step, you really need to think about your initial hypothesis of what's causing bacteria. And to do that, you accumulate your data. For example, fecal bacteria levels at each site that you're monitoring, getting insight from local partners as far as what can be occurring, finding out the age, location, condition of sewage and septic systems along the watershed, land use within the watershed, sanitary surveys, and field reconnaissance. So all these things you start putting together and getting your initial hypothesis, saying, okay, you know what, the obvious things that could be causing pollution in my watershed are, well, we have no agriculture, it's a beach, there's discharge from inland residential, and so it could be dog, it could be human, it can be gull. That would be one scenario. And this is all based on existing lines of evidence, really looking at the low-hanging fruit of what you know about your watershed before beginning one of these studies. And it'll help guide you as far as what you're going to be looking for with the more expensive analytical methods. From there, three elements are sampling sites. So selecting sampling sites, that tends to be fairly straightforward. Since most people are monitoring anyway, you simply go to your hotspots, where you have high levels, where you've historically had high levels. If the hotspots are not that obvious, then collecting near physical sources close to agriculture, close to residential, where you know that physically these could be potential sources. are a good strategy. Then sampling events. Wet versus dry weather sampling tends to be regulatory driven in that most people have a period of time or condition when they're monitoring for their, let's say, MS4 discharge permits or whatever else you're monitoring bacteria for. But then there's also seasonality. In a lot of studies, we've observed that seasonal changes do occur in pollution sources in bacteria levels. And so something that you should be thinking about when setting up the number of events. And then finally, it's very important that you have statistically significant number of events. A lot of times we've worked on projects and there just simply weren't enough sampling events. And so that one shot you spend all your resources on, and there was no pollution, there was no bacteria, and so the source identification is a moot point when you have no bacteria to begin with. And so you want to make sure that you have a meaningful number of events to interpret what's going on within that watershed. Remember, these temporal differences are significant, and sometimes they're the most significant factor in certain cases. So sampling events, those are things to consider. And then the tests that you're going to be running on each sample. What the California guide document recommends is really focusing primarily on human, because if human is there, everything else doesn't matter, right? But then going beyond human, you want to look at anthropogenic sources, things that you can control: human, dog, agriculture. The sources that are controllable, that you're going to be able to do something about. And then from there, if there are no controllable anthropogenic sources, then it is helpful to look for the wildlife sources that you believe are the primary cause. If you think it's bird, if you think it's gull, deer, some other kind of wildlife source is the primary cause, and you have good data showing there is no human pollution, then you can go in and start testing for these wildlife sources. So you have a healthy set of data saying there was no human fecal indicators, and then a complementary set of data saying, and there was a high degree of bird fecal indicators in our samples. So from there, when you're collecting the samples, you would, for microbial source tracking, use the standard procedures that you would use for collecting your normal bacteria monitoring samples. We ask for a bit more water, usually 500 milliliters, so that we can concentrate higher volumes of higher levels of bacteria. And so the water samples can be simply collected like you would normally do, put in a cooler like you normally would do, and then shipped to our laboratory overnight. And because these are genetic methods, the hold time is not as restrictive as the enumeration general bacteria monitoring methods. And so we receive a good number of samples as water. The way many big projects are doing it now, though, is that you can have the local laboratory that's doing your routine monitoring filter the water samples. And so these pre-filtered samples can be stored, frozen locally, and you could take your, whatever it happens to be, 100 samples a year, filter a subset of that water, and then store it. And then you say, okay, well, we only frankly have the money to test 20 of these or 30 of these samples with the more expensive source tracking tests. And so you can go in and cherry pick which samples are going to provide you the best insight in terms of high levels of bacteria. So you say, okay, we realize these 4 sites this year were paired. Let's take those 4 sites every collection event and send them for analysis. And let's figure out what's going on there. So it frees you from having to send continuously the water samples and enables you to go back and determine which samples you're going to analyze with the source tracking markers. So the results of microbial source tracking, On the left column, this is an example report, and what source tracking provides you are essentially two things. It tells you in a sample, was there human or not, was there dog or not, was there bird or not, was there any of those sources we tested for, were they present or not in the sample? That's the first element. The second element is, okay, it was present. What was what was not necessarily the relative contribution, but what was the magnitude of that source in my sample? Was there a high signal strength, a strong signal of human fecal indicators, or was there a low signal? And then on the right column, set up an example set of results from, let's call it site 1, 6 sampling events. So each one of these samples is taken, let's say, from one location over the series of 6 different days. And what these bubbles represent, red being a positive result, negative being represented as a gray result. The size of the circle would represent sort of the signal strength or the quantitative value. And so what we see here is pretty consistently we're finding a lot of reds for human, not a lot of reds for dog and bird. And then the size of the circle is getting bigger. You see that these human results are pretty big. So this means that you have a high frequency of positive results for human, and the circles are big. So that means that not only was it present, but it was present in a pretty significant concentration in each one in most of these samples. Now you see an example here. There is one positive for dog. It could mean there potentially is dog there also. Now, can you tell exactly how much was from dog and how much was from human? No, you can't. But what you can see from this example data set is that human is most likely the significant contributor to fecal pollution at this site. And so this is really the way to interpret data from microbial source tracking analytical results. It's not going to tell you proportionally how much pollution is coming from each bird, human, and dog, because they're targeting independent fecal indicators that don't necessarily relate to one another. But what it's going to tell you is that in this case, you probably have a human problem. And so, I'm emphasizing this because a lot of people misunderstanding is that they're going to get the original pie chart idea from over 10 years ago, which never really worked out. But what is working is this form of interpretation of data. So how do you actually interpret what's going on in your watershed overall as a project? Well, it's important to factor everything in, not just the raw analytical data. And so this is a scenario 1, a site with high fecal bacteria, potential sources, human and goal. Well, the weight of evidence is pointing towards a non-human pollution source. So what's going on here, you have fecal indicators present, but only during wet weather, which means it tends to be stormwater runoff, surface runoff, that's causing the high levels of bacteria. You have the presence of goal fecal indicators in the series of microbial source tracking samples that were submitted. You have high concentration of those goal fecal indicators in the samples that were submitted. So not only do you have a lot of positives, but you have high concentration when they're positive. And then finally, your sanitary survey showed a systemic presence of goals on the beach. What do you have on the human source side? Well, you have actually an absence of human fecal indicators. So you did a series of sampling events. You didn't find human fecal indicators, and your sanitary surveys showed no evidence of leaking source. So this is really a very clean and simplified way of interpreting a study, overall study, incorporating the analytical results with the lines of evidence that you have. And so in this scenario, it's most likely that you have a non-pollution, non-human source, and not necessarily a major human problem at this site. So let's see scenario 2. Scenario 2, another site, a different location, maybe inland, high levels of fecal bacteria, potential sources human, bird, and cattle. On the left column, you have the absence of any bird fecal indicators. You have the absence of cattle fecal indicators. On the right, however, you have the presence of human fecal indicator 1. So you can do different fecal indicators, unique indicators. And so fecal indicator 1 was present in the vast majority of the samples. The concentrations, the quantification values were very high. You added a second fecal indicator, let's say in this case the US EPA developed fecal indicator. Again, the concentrations were high frequency of positive hits. Sanitary survey showed aging infrastructure. Your fecal indicators are present during dry and wet weather, which might indicate you have a leaking sewer system that's constantly introducing bacteria into the water system. So in this example, scenario 2, you're putting everything together, and the weight of evidence is pointing towards human as the primary cause of your elevated levels of fecal bacteria. The source tracking methods and tests are really meant to be a tool to guide you with other lines of evidence, and not necessarily just a standalone analytical magic bullet. But they play a very important role in how you put these studies together. So what does an actual result from a study or an outcome look like with microbial source tracking? Well, we have 3 case studies.
As summer approaches and water fun is on everyone's mind, one county is worried about more than just the water levels.
Yeah, they're really worried about something pretty nasty in local rivers. Devin Neely has more.
It's starting to warm up outside, and rivers like the Animas can be a nice place to cool down. But there's something in the water that might not make that a good idea.
The state has found over the last several years that there's been E. coli levels that are too high for safe recreation. And so there's been kind of a lot of speculation about where that bacteria in the river is actually coming from.
Over the last year, the San Juan Watershed Group has been trying to identify a source for the bacteria in the Animas and San Juan rivers.
It was actually quite surprising. We found a lot more human sources than we were expecting.
While the San Juan Watershed Group is careful to point out that the results are preliminary, they say the problem could be leaky septic systems, and San Juan County is already working on a solution.
Expanding sewer out into the unincorporated part of the county and getting folks off of septic systems would be ultimately beneficial to the surface waters.
But in a county through which 60% of the state's surface water flows, is the water safe to play in?
Today, I think I would. We didn't see too many. High bacteria levels early in the season last year, but for the most part, the most dangerous levels of bacteria we saw were when the river's really high, really muddy. I definitely wouldn't go swimming on that day.
Devin Neely, KOB Eyewitness News 4.
Scary stuff there, and that study will continue through October. Once they get more information, the Watershed Group will work on ways to clean up the river.
This San Juan watershed study was one of the big, ambitious studies that we've been doing over the last two years, and we feel really proud about the outcomes. They had expected to find very little human fecal pollution. Surprisingly, they did find pretty significant levels, and as a result, they're going in, improving the community's water quality, and making it safe for the residents to recreate along that river. And so this was a pretty large project, a few hundred samples over the series of 2 years. So the next case study is a project that we did along with Geosynthetic in the San Diego River for their MS4 permit. And so this schematic here kind of outlines the flow of how source tracking can be applied on MS4 projects. And so the first step, looking at your data review, evaluating your GIS information, doing an observational investigation of what's going on. Do you have flow or do you not have flow? And if finally you do have flow in your discharge, then you go about testing for human fecal indicators. If those fecal indicators are present, then you need to go in and try to investigate up the water storm drain system to hone in physically where you're finding human pollution. So that would require additional source tracking within the actual storm system. Finally, you mitigate the flow from the key areas in the discharge where you're finding source tracking markers, then do verification monitoring for human markers, again, and fecal indicator bacteria until you find no more human pollution. And then if you do get to that point where you finally were able to address all human fecal indicator sources and you still have flow, then it is potentially possible to apply for a QMRA, which is an alternative monitoring limit based on the fact that you show there is no human impact on your water system. And so it's a fairly complicated schematic, but it gives you an idea of sort of a process framework of applying source tracking on stormwater outfalls. And I think it's a very useful tool guiding decisions on using this method for stormwater discharge. And stormwater discharge has slowly become the leading use of microbial source tracking across the country, at least from what we've observed. And it's a very interesting tool for stormwater discharge permittees. And so the final case study is a project where we're going to go into a little bit more detail of how an actual project is structured. And so this was a project in Delaware on a beach where they were observing high Enterococcus levels. So they gathered their information. Their existing lines of evidence showed visual observation of dogs at the beach. They had large concentrations of gall at the beach, and there was a geographic proximity to septic systems that could potentially causing problems. So their hypothesis was that gall, dog, or human are likely sources of their high bacteria. Their sampling plan, they went along the water system, found 3 locations where they were finding high levels of bacteria. So they collected 3 samples per week for 3 months. So they took 3 water bottles along the beach, combined them, and then did that 3 times every week, totaling 49 samples over that 3-month period. And so they chose to do 2 unique tests for human fecal bacteria. They did 1 dog fecal indicator. They did 1 gall fecal indicator, so a total of 4 tests. The samples were filtered locally, frozen, and shipped on dry ice. And so the results of that study were that human fecal indicators were absent 92% of the time. So the vast majority of the time, there were no fecal indicators in the samples. And so the very few times when the fecal indicators were present, the concentrations were very low. So you have a low frequency of human fecal indicators, and even when they are present, you have a low concentration. Dog, very similar results, even fewer positives, and again, when I believe even just 1 sample was positive, in very low levels. Gall, on the other hand, 100% of the samples were positive for the gall fecal indicator. And so every sample had the gall fecal indicator, and the abundance was very high. So they were significant. There was a significant abundance of this gall fecal indicator in their water, in their watershed. And so, based on their existing lines of evidence from their sanitary surveys, combined with the genetic qPCR results, they concluded that gall was most likely the major contributor to the elevated levels of Enterococcus at this beach. And so because of this, what I understand is that they were able to shift resources to other locations which were more likely to be impaired by human fecal pollution. And so this was pretty significant cost savings for that community, since they could have jumped the gun and made pretty significant investment in infrastructure improvements, and that was not really going to address the problem. They could have spent millions of dollars putting sewage lines into that community, and those levels would have still been high in spite of that investment. And so this can be a powerful tool in locating resources within a community, which has an even more significant impact, with budgets being pretty restrained. And so that's the presentation. We went over the development, how the method's used, and case studies on how this technology has been applied across a series of different projects. And so now we can open it up to questions. There's contact information from myself and Grace, and I thank each of you for attending.
Mauricio?
Yes.
Yeah, it's Eric. I sent you two different questions. The first is related to slide 11. Mm-hmm. And the question was, what do you mean by significant number of samples needed? What type of range are you trying to indicate is needed here?
Well, that's a discussion. Actually, frankly, that's a discussion between the watershed manager and the regulatory body. We do projects as simple as a handful of samples, and they were meaningful because they were just like discharge from a courthouse, through the roof E. coli counts, and they sent us a handful of samples. They were flaming hot for human, and the lab thought they just sent us sewage. And they went in and they found the broken pipe. So in that case, just a handful was fine. That New Mexico project, I believe it was, a few hundred samples. I think it was about 200 or 300 samples. In that case, it was a complex water system. They had many different locations across miles of property, of land. And they had many different sources. They had conditional factors. In that case, they needed to do a few hundred samples. So it's really dependent on the goal you're trying to achieve. And maybe I can give you real numbers for the other case studies. The Geosynthetic study, the San Diego study, I believe it was, just a few dozen, maybe I think less than 100 samples. And their goal there was simply just take 40 discharge points and let's see if there's any human at any of them. And then do a follow-up study on the ones that were positive and go up the water system. So it's going to vary. It's going to vary, and it's really a discussion to have on a case-by-case basis.
Okay. Another comment was sent in. If you can check the chat that I sent you, Mauricio, it's a paper.
Yes.
Do you have any comments on that? For those of you on the line, it was Enterococcus and E. coli fecal source apportionment with the microbial source tracking genetic markers. Is it feasible? And Mauricio's answer is yes.
I mean, I don't know the study offhand. I mean, I'm actually just pulling it up now. This one, I'm going to have to make a 2-second assessment of a big research study here, but let me see if I'm familiar with it, and I can give you an answer. There's an attempt to do source allocation. Oh, okay. Yeah, Dan Wang. I don't know. I don't think they were able to make it work in the evaluation. I don't think they were able to make it work. It's not impossible, but what happens is the simple limitation is that you can throw a gram of human sewage and a gram of dog fecal material into a river, and it'll work probably that first second after you drop the samples into the water. But what happens after a day? What happens after a week of that bacteria being in the water? I mean, the decay rates are going to vary. And so that's what I've seen for a location, is that if you can capture it the moment it dropped in there, it'll work. But that's not really realistic. And so we've shied away from it. I think it's going to be possible at some point in the future, but we've shied away from it. That's my opinion.
Okay, I have a couple more that have come in.
I see one here about wildlife, ducks, wetland, rice, discharge concern, deer, or other upper watershed pathogen issues. So there's a test that could potentially detect duck. Deer also, there's a test that can potentially detect deer. Those are two of the trickier sources.
Hey, can you talk about the differences between available human markers?
Yeah. So what happens is you have thousands of different kinds of fecal bacteria in human sewage, right? And so within those fecal bacteria, you have millions of different genes, and so they're just unique. They're unique, and one gene might be present in one set of sewage more than the other gene. They're just unique genes, and so by doing two different genes, you're increasing your confidence, because you're going to find that one gene maybe in higher concentrations than the other gene in some examples, and one of them is like the original source tracking qPCR gene, and the other one is the US EPA developed and licensed gene. Those are the two most commonly used ones. Those are the two recommended in the California guide document, and usually it's good to do both genes, especially even in septic, where you don't know how much of each of those genes are in the starting sewage. I hope that answers it.
Okay, another question for you. Are you also looking for antibiotic resistance?
No. That was sort of the approach a while back, and it was interesting in the California validation study that 27 labs took part in. Not a single lab chose to test an antibiotic-resistant method across the entire country. So that sort of gives you an idea where researchers are today on that form of source tracking. I think you really moved and invested into this qPCR. And in fact, which is, let's say, antibiotic resistance is like generation 1 of source tracking. qPCR is, let's say, generation 2. Digital PCR is 2.5, and generation 3 is going to be, next generation sequence, community analysis, 10 million data point, computer interpretation things that are going to eventually replace qPCR, but that's still pretty far out there.
Okay, we have a question. Does digital droplet PCR and/or traditional qPCR provide the same levels of variance that are seen with cultivable methods?
Okay, let me. So variance. I mean, if you're talking about concentration variance, like if you take one sample and then you take another sample 5 feet away, and you're going to find really varied results. If that's the variance you're referring to,
well, this is, I guess, variance from the method.
Oh, I mean, yeah. I'm just trying to make sure I'm answering the right variance. Maybe if I just answer both. Variance across two samples 5 feet away or 10 seconds from each other, there's inherent variance in collection of water that's going to exist whether you're culturing or doing qPCR. Variance from older culture-based source tracking methods, this has reduce significantly the variance in the results compared to the older methods. There is a question here. I see the current cost. So we're charging a few $100s. It's going to depend on the number of tests that are being done. So in the best case scenario, maybe like $130 per test if you're doing a lot of tests on a large number of samples. But then if you're just doing like one sample with one test, it's going to be a few $100s for that single sample. But it's heavily project size dependent. There's a tremendous efficiency we can gain by doing high volume projects. And comparison. Okay, can you please talk about current costs, including comparison of digital PCR and qPCR? Well, qPCR is theoretically cheaper because people have the equipment already. Like us, we already have the equipment. Digital PCR has this big capital cost, upfront capital cost. But then in the long term, what I understand is that the per sample cost goes down. But you have to pay for these big expensive machines at first. So we're charging about the same for larger projects.
Okay, we have a question sent in. One of our participants was hoping for a bit of advice on selecting which would be an appropriate, method for specific sites. And is that handled also in the California guidance?
They have recommendations of tests for the specific sources that might be causing pollution at your sites. But we do help. I mean, we sometimes work through an engineering firm that provides insight on structuring a project, but we do provide assistance also on, whatever knowledge we've gained over the last 12 years, and we do try to help people in more of a supporting role on how to set up these projects and which tests to use. And there is advice in the guide document.
Okay, thank you. Next question is, what is the general marker used? Referencing the slide which showed the report for human-specific marker.
General marker, hold on. I'm not quite sure which slide that is. If you get a slide reference. Yeah, I'm not quite sure. What do you know what slide that is?
There was no slide number in the question. It's just what is the general marker used, referenced on slide showing result report for human-specific marker.
Oh, okay. Yeah, this one. That's the GenBac 3. That's usually there's like an EPA standard for general Bacteroides, and so we use that standard assay. I have here another question. Talk about the specificity of the canine test. Does it capture domestic dogs, coyotes, foxes, or raccoons? We've validated, I think once or twice, one of the older, not the EPA marker, but the other dog marker, and it did come up positive in coyotes. How often does that happen? It'd be really hard to say with just 2 data points, but we have seen it. And the EPA marker, it just was published in September, so we have pretty limited data on that one. And there was another question here. Do you know when the EPA will publish, approve an official method for source ID? No clue. The bureaucracy at the EPA is pretty impressive. I mean, just to get our license took a few years. To get a license and an update for the dog marker. I mean, these things take their time. It's anyone's guess. But I think the data and the submission to the bureaucracy machine should be done this year, and then how long it takes to churn something out from there is anyone's guess. But hopefully in the next year, if you want to be optimistic. Can you please talk about the specificity of canine test? Oh, okay, I answered that question. Can you distinguish between cows and deer? Yes. Yeah, so the EPA test has the capacity to discriminate between cows and deer, both being ruminants. So we have a ruminant test that will pick up either one. And then we have the EPA cow test that will pick up only cattle. So the short answer is yes.
If you can also ask Mauricio your questions, just hit *6 on your phone to unmute and ask a question. You can continue to send questions via chat as well if you like. Thanks for sending all these questions. Appreciate it.
May I ask a question?
Yes.
Okay. My main interest is, Dr. McGowan, my main interest is in antibiotic-resistant bacteria and their genes. And the question I would ask is, how do we contact you? Let me give you my email address. It's edo_mcgowan@hotmail.com. How do we interconnect with you and others who might want to do these tests but find no reason to do them economically? We are losing our antibiotics medically, and as you indicated, there's no general interest in tracking these things, and the question is why? This is a national crisis. So if we open up a conversation offline, that would be wonderful.
Yeah, sure. I think we can continue this offline. I'll have your email so I can send you a note with my contact details. Thank you.
Okay. Thank you so much.
We have one that's about testing felines. People are seeing cats in their creeks.
Cats. We don't have a cat test. I saw something the other day. Theoretically, there is a cat test, but I haven't seen a lot of work in that area. I mean, I think it's an area of interest. There probably will be, but my understanding there is no cat test right now.
On behalf of Source Molecular, it is indeed my pleasure to express gratitude to the California Water Quality Monitoring Collaboration Network, and special mention to you, Eric, for hosting this webinar today. And I wish that this presentation could add significant value to all of you in your objective to address your fecal pollution problem. So once again, thank you, everybody, for being with us today.